Networks form an important part of our lives, from the social networks that connect us to our friends, to the streets we navigate on a daily basis. It's currently unclear how the brain processes the information contained in these networks. One candidate brain region that might do this is the hippocampus. This brain region is thought to contain an internal map of space that helps us remember the past, but also imagine the future. Neurons in the hippocampus, known as place cells, signal an animal's current location in the internal map through their activity patterns. Recent research has shown that place cells also contain information about how different parts of the environment are connected. To explore how the human hippocampus might represent the structure of the environment, we recorded fMRI activity from participants as they navigated the streets of London's Soho, for which they'd been trained the day before. Navigation was tested by showing movies in which participants were first informed where they were, then by giving them a goal to get to and asking them is it on their left or right. Ahead of each street junction, participants had to decide which street they would take to reach the goal, after which the movie took them along the route, pausing to probe their decisions. To examine how the brain processes the street network, we extracted information about the structure of the environment and collaborated with researchers who use a method known as space syntax.
Space syntax is a method which allows us to apply graph-theoretic measures to a street network, treating the street segments as nodes in the graph. If we take an example, imaginary street network, one of the measures, degree centrality, measures how many streets are connected to that street. Here we can see the one street that is highlighted in red is connected to 6 other streets, while others have fewer connections. Another measure is closeness centrality. This measures the shortest number of streets needed to travel through to reach all other streets in the network. Here we can see some streets in red are topologically closer to all other streets in the network. Another measure is betweenness centrality. This measures the number of times that a street would be travelled through for all trajectories in the network. We applied these measures to central London in the United Kingdom. Here we can see betweenness centrality for central London, and for closeness centrality, and finally degree centrality. If we zoom in on Soho, in the white region, we can see how the pattern of degree centrality varies across the streets.
So we took space syntax measurements and entered them into our analysis of fMRI data. We examined both raw values and the change in values.
In an example route, we can see the changes in degree centrality along this route. In this case, the degree centrality decreases after the first 2 turns and then rises again towards the end of the route. We found that activity in the right posterior hippocampus exclusively tracked the changes in degree centrality. This only occurred when a new street was entered and while navigating the routes. It did not occur when participants were told which way to go in our control task, and was not explained by visual properties of the streets. In addition, we also examined the brain regions involved in searching the future streets during planning. We measured the number of possible future paths available beyond the next junction. For some junctions, this was low, for others, it was higher. Activity in lateral prefrontal cortex was greater when there were more streets to consider.
This study is important because it gives us critical insights into how the hippocampus and the prefrontal cortex might operate together in a task as simple as navigating our way through a city street to a goal. We didn't previously know when or which brain regions might be doing this kind of simulation of the future to allow us to decide, do I go left or do I go right to get to my goal? Now we have some insights in this study, and now we can hopefully move beyond to look at how precise cells in these regions might operate at a more fine-grained timescale.